So I'm sorry, I got logged out by network. So today we are moving to study section 4, which is the general characteristics of microorganism. So in study section 3, we treated general characteristics of microorganism, which is I think we treated bacteria. Yes, so today we are moving to virus. Can you hear me now? Some people said they can't hear me. So in case you stopped hearing my voice, that means network has locked me out. But now, am I audible enough? Some people said they can't hear me. Am I audible enough? Can you see the full screen now? Okay. All right. So now let's move to general characteristics of microorganism, virus. So because of our time, I will not be reading line by line. So let's just move to virus. As nurses, we have been working in the hospital. Some have had like 10 years experience, 20 years experience, and medical microbiology. Microbiology is not something new to us. Although medical microbiology might be new, but in School of Nursing, we did microbiology. And even in secondary school, in our biology, we did microorganism, bacteria, virus, fungi, protozoa. So today, we are talking about virus. And why do we need to know? Because we are nurses. We know we have a lot of viral infections. So we need the knowledge of all these microorganisms, so that we will know how to prevent them. You need to know their characteristics, their mode of replication, things that are peculiar to all these viruses, because we have a lot of disease conditions that are caused by virus. So that is why it is important for us to know the characteristics of all these microorganisms. So what is the definition of virus? A virus is just an infectious microbe that consists of segments of nucleic acid. So the nucleic acid can be DNA or RNA. And we have different, it can be double-stranded, it can be single-stranded. But it is just an infectious microbe that consists of nucleic acid, either DNA or RNA. And it is surrounded by a protein coat. And viruses are also called virion. Viruses are also called virion. Now let's go to general characteristics of a virus. Viruses are made up of genetic material. I just said that now when I was defining virus. So they are made up of genetic materials, which might be RNA or DNA. And it also has a protective protein coat known as capsid. So it is this capsid that is protecting the outer layer against any environmental temperature, against any form of harm or danger. So it is this particular capsid, is a protective protein coat that covers the virus. So they have another layer that is called an envelope around the capsid. So viruses without an envelope are called naked viruses. Take note. They are called naked viruses. So they are similar to parasites because they need a host to reproduce. A virus cannot just live on its own. They cannot survive on their own. They need a host to survive. So it is just like a parasite. So they need to depend on host to feed. They need a host for them to replicate or to survive. So they will survive outside of a host until their capsid breaks down over time. Now, it is about 100 to 1,000 times smaller than the cells in our body. So if they are smaller, they are very small. When you compare virus, the size of a virus to that of bacteria, it is very, very very, very small. I hope you are following me. All right, so this is the diagram of a virus. So can you see the diagram of a virus on the screen? So the last time we treated bacteria, I also showed you the diagram. So this is the diagram of a virus. So can you see? It has a spike. It has a capsid. That capsid is the protective outer layer, which is surrounded by envelope. It also has a spike. It's this spike that helps the virus to attach to its host. Then it has DNA. They have DNA, RNA. Now let's talk about the structure of a virus, one after the other. So the capsid. The capsid is the protein shell that encloses the nucleic acid. So it is this capsid, I've told you that it's the outer layer, and it's a protective what? Protein coat. So it's the one that encloses the nucleic acid. So with its enclosed nucleic acid, it is called nucleocapsid. So nucleic acid. So capsid together with enclosed nucleic acid is known as nucleocapsid. So this shell is composed of what? Protein organized in subunits known as capsomers. So this capsid, the shell, is composed of protein organized in subunit known as what? Capsomer. And this capsid has three functions, three main functions. I've told you one of the functions, which is what? It offers protective function. Then it protects the nucleic acid from digestion by enzymes. Then another thing is that it contains special sites on its surface that allows the virion to attach to its host cell. So this capsid contains special sites. One of it is the spike. So it is this special site that it uses to what? To attach to its host cell. Then another thing is that it provides protein that enable the virion to penetrate the host cell. So these are the three main functions of this particular capsid. It protects the nucleic acid from digestive enzymes. It contains special sites on the surface that allows the virus to attach to its host cell. Then another thing is that it provides a protein that enables the virus to penetrate its what? Its host. Now let's talk about the envelope. Many types of virus have a glycoprotein envelope that surrounds the nucleocapsid. So the envelope is composed of two main lipid layers that are interspersed with protein molecules, and they contain material from membrane of the host cell, as well as that of viral origin. Then another structure we want to talk about is spikes. I told you that these spikes helps them to attach to their host cell. So it is this particular spike. You can see that in that diagram, it looks pointed. It looks pointed. So it is with the help of this spike that helps them to what? To attach to their specific cell surfaces. Then now nucleic acid. So the nucleic acid, we have both RNA and DNA. So the nucleic acid of each virus are the ones that encode what? They encode the genetic information for the synthesis of all protein. So DNA is just like an instructional manual for how to build the virus. You know, they need this to replicate. Very, very important. Although they cannot replicate on their own, but they need this RNA and DNA to what? To reproduce. So it is DNA that is like an instructional manual for how the virus is going to what? To reproduce. So RNA is just like the translation of the instruction in a language that the cell machinery can read and now make into proteins. Please, can you still hear me? Okay. So now let's go to classification of viruses. Classification of viruses. We can classify viruses in three major ways. The first one, they can be classified according to their shape and size. The viruses can be classified according to their what? Genomic properties. Then another mode of classification is what? Is mode of their replication. So these are the three ways you can classify viruses into shape and into shape and their size. They can be classified according to their genomic properties. And another way is mode of replication. So we are going to talk about all these one after the other. So let's go to classification according to shape. So viruses have different shapes. They have different shapes. So one of the shape that we can see is icosahedra or polyhedra. So one of the shape of a virus is icosahedra or polyhedra. So this is a geometric shape with many sides. So this polyhedra, you can hear the word poly. Poly means many. So this is a geometric shape with many sides. So when you see this type of virus under microscope, they look like a soccer ball. Exactly like what? A soccer ball, because they have many sides. It has many sides. So we refer to them as icosahedra or polyhedra because they look like a soccer ball. So they have many what? Many sides. Then we have the helical. These virus shapes look like a cylinder. So this helical looks like what? A cylinder. They are cylindrical in shape. Then its genetic information is coiled up like a spring inside. Then another shape that we can see in virus is what? Spherical. Spherical shape. So these spherical viruses are helical or polyhedral viruses that have an envelope around them. So they look like a ball. So from the word spherical, that means sphere, it is round. So they look like a ball. Then lastly, we have complex. We have complex. Complex viruses combine more than one shape. They combine more than one shape. You can see more than one shape on a virus with complex shape. So I hope I'm making sense. So now let's go to classification according to what? Virus sizes. I told you earlier on that viruses are very small. They are small, very, very small. So we are not really going to talk much about it. Just have it at the back of your mind that they are small, very, very small. So all viruses are very small. They are too small to see without a microscope. It is not possible. We have to use a microscope before we can have a view of what virus looks like. So if you measure them under a microscope, most are between 20 nanometers to about 400. So you can see that they are extremely small. When you hear the word nanometer, they are what? They are very, very small. So for comparison, the smallest virus are about 2,000 times smaller than a grain of sand. You can imagine. They are about 2,000 times smaller than a grain of sand. And they are about 100 to 1,000 times smaller than the cells in our body. But their sizes can vary a lot. It can vary a lot. Take, for instance, the measles virus is about 5 times larger than Zika virus. Viruses also have varying weights. So have that at the back of your mind. So classification according to what? Their genomic properties. This is the most important part. The genetic material of the virus may contain DNA or RNA. And this DNA or RNA might be single-stranded or double-stranded. It can either be single-stranded DNA, double-stranded RNA, depending. So it can be single-stranded and it can be what? Double-stranded. So DNA viruses can have their genetic material in a single string of instruction. You know, it is this DNA that carries the what? The genetic code that carries all the necessary information that the virus requires to what? To replicate themselves. So that is just what we need to know here. So these viruses can have their genetic material in a single string of instruction or two sets that are paired together. That one will be double-stranded. So if it is a single string, if it has a single string, that means it is single what? Stranded. But if there are two sets that are paired together, that means it is double-stranded. It is double-stranded. But RNA viruses are usually what? Single-stranded. Though there are some double-stranded RNA viruses. Are we still together, please? All right. So now let's go to classification according to what? Mode of replication. How do they reproduce? So we classify them according to their what? Mode of replication. Now, viruses have several steps to infecting cells and reproducing. You know they cannot survive on their own. They need hosts to reproduce. They have to attach to a particular host. So their mode of replication has different steps. The first is attachment. Then another stage is what? Entry. Then we have replication, assembly, and release. So please take note of all these what? These steps. They have several steps that they have to what? To go through in order to infect cells and reproduce. So the first stage is what? Attachment, entry, replication, assembly, and release. So please take note of all these 5 steps that they need to go through. So let's talk about the attachment and entry. Let's talk about the attachment and entry. For attachment and entry, from the word attachment, that means they need to attach to their own cells. So they can get into the cells in 3 ways. So viruses can get inside the cells in 3 what? Major ways. That is under attachment and entry. So the first way by which they can attach and enter into their host is through receptor binding. Receptor binding. So what happens at this stage of receptor binding? So what they do is that cells, you know that cells has receptors on their outside, externally. So these receptors, now what these viruses will do is that these cells have receptors on the outside that can receive signals from proteins in our body. So these receptors are just like a door. The receptors on the cells, they are what? They are like a door. So viruses will now trick the cells into thinking they should be allowed in. So it is like a door. So it is these receptors that they will bind or they will attach to. I don't know if you are getting what I'm saying. Please, are we together? Please, am I making sense? Please, let's respond. Okay. So that's the first way by which virus can what? They can get into their what? Also through receptor binding. So they bind to the receptors that are outside the cell. Then another way is through direct fusion. So that one, they just attach directly to the host cells. They will attach directly. There is no further explanation on that. Then another way is through bacteriophage. They will inject, bacteriophages inject their genetic material into bacterial cells, and the entire virus doesn't need to get inside. So that is the third way by which a virus can get into their host cells. So that is another way by which they can get into their host cells. Now, the second stage of replication, the second stage, or let me say the second step that we are talking about is replication. We have talked about the first step, which is what? Attachment and entry. Now, the second step is replication. So what happens in this step of replication? The viral genome contains all the information necessary to produce new viruses. So once inside, the virus induces the host cell to synthesize the necessary components for its replication. So that is what happens at the replication, at this step of replication. Are we still together? So virus cannot reproduce by themselves. So as the host cells is replicating, they will be replicating together with it. So that is what happens in replication. So now once the virus or its genetic materials inside of a cell, it now uses either a lytic circle or lysogenic circle to reproduce. So once they find their way into their host cell, they will now use either a lytic circle or a lysogenic cycle to reproduce. Then we have some viruses that also uses both this lytic circle and the lysogenic cycle. So immediately they find their way into the host cell, they cannot just start reproducing. Something must definitely happen. So they either reproduce by means of lytic cycle or by means of lysogenic cycle. So we are going to talk about those cycles one after the other. So in the lytic cycle, what happens? From the word lytic, lysis means what? Breaking down. From the word lytic, that means breaking down. So just think about that, breaking down. So what happens in lysis is that the virus uses its host cell's machinery to make copies of itself. So in this stage, the virus cannot, they are the impatient type of virus. Immediately they get into their host cell, they become so impatient and they begin to replicate. They begin to use what? The old cells machinery to make copies of themselves. And they get, it gets to a point, their number increase to the point that the host cell will now burst. So pieces of the viruses assemble, wrapping up the genetic material in the capsid. So viruses make many copies of themselves. So eventually we have so many copies of this particular virus, and the cell now what? Burst. So if there are any surrounding cells around that place, this virus will definitely what? It will affect them, or it will infect that particular cell. So maybe I should come again. Lytic cycle, what happens? The virus use the whole cell machinery to make copies of itself. They rely on the whole cell machinery to begin to continue to make copies of themselves. And it will get to a point, the copies will get so large to the extent that the cell will be forced to burst. So that is why this stage is known as lytic cycle. So they make so many copies of themselves to the fact that the cell can no longer contain them. So the cell will be forced to burst. And this virus will escape from that particular cell and move to to the surrounding cells. So that is how they continue to infect other cells. So now let's talk about the lysogenic cycle. So what happens at the lysogenic cycle? So this lysogenic cycle is different from the lytic cycle in the sense that the virus does not replicate quickly. So here, these viruses remain dormant or silent. They are not in a hurry to replicate, unlike in the lytic phase, whereby they continue to make copies of themselves using the whole cells machinery. But in lysogenic cycle, this does not happen. The virus just stays there. The virus will be silent. The virus will be dormant, waiting for the proper time where the cells, normally cells in the body, continue to reproduce. So this virus will just lie there silently, doing nothing. And it will wait for the time that the cell will reproduce, and it will begin to know automatically this virus is already part of the cells. So automatically the cells will make copies of that particular virus. So let me read what I have here. So viruses are dormant, they get inside the cells and then wait. So I made an illustration here. I said instead of setting up a shop to cook in your kitchen right away, it is as if they put their recipe into your body's recipe book without you knowing it. So they just stay there, they are waiting. So the cells don't realize that the virus is there, and then they will continue to reproduce as they normally will. The cells will continue to reproduce normally without knowing that, there is a particular virus that is lying down there, silent or dormant. So each new copy of the cell, as the cells, make a copy of itself, it will continue to make the copy of of the virus along with it. So that is just the simple thing that happens in the lysogenic cycle, unlike the lytic cycle that, it cannot wait. In the lytic cycle, the virus does not wait for the cells to reproduce. All it does once it gets there, it becomes so impatient, it will just wait, it will just be there and continue to make use of the host machinery to make a copy of itself. It doesn't wait for the cells to reproduce. It doesn't wait. But in lysogenic cycle, that doesn't happen. The virus just waits for the cells to reproduce, and then it will make a copy of the virus. So I hope I'm making sense. Any questions so far? Please, let's respond. Okay. Same question. Yeah. Someone is saying according to chemical morphology. Please. Okay. I'm trying to read your questions. There's nothing special about shape. I told you it is just straightforward. We have different types of shape of virus. So it is straightforward. Not There's nothing special about it. I told you we have the complex, we have the spherical, we have the icosahedral or poly. That one looks like what? From the word poly, I told you it has many sides, just like a soccer ball. All you need in that particular paragraph is for you to know the different types of shape of bacteria or virus that we have. Once you hear the word polyhedra, that means what? That means it has many sides. Or icosahedra, it has many sides, just like a soccer ball. Once you hear spherical, that means it is round. Complex, you can see more than one shape. So that one we cannot say, okay, we cannot say whether it is spherical or it is icosahedra or it is complex. You can't say. That is why they call it complex. That is why it is called what? Complex. Nobody can say. You can't even say, okay, this is the type of shape that that particular virus has. So that is why it is being named as what? Complex. Then for lysogenic cycle, please listen again. I said in lysogenic cycle, once the virus gets its way into the host cell, you know that normally our cells, reproduce from time to time. So when this virus gets into the host cell, it doesn't wait for the time that the cell will what? That the cell will reproduce. It doesn't wait. It doesn't have a patient. That is in the what? In the lytic cycle. So it will continue to make use of the host machinery to make a copy of itself. That is what happens in the lytic phase. But in the lysogenic phase, this virus will lie down there silently. It will lie down there silently. So it doesn't rush. There is no rushing. So as the cells make a copy of themselves, what will happen? They will also be making a copy of the virus. That is just a simple illustration. I don't know how to explain better than this. That is the difference between the lytic cycle and the lysogenic cycle. This virus will be there. It will lie down dormant. The cell will not be aware that, oh, something is present, virus is present. So as the cell is making a copy of itself, it will also be reproducing the virus, because the virus is already a part, or let me say component of that particular cell. So as that cell is reproducing or is making a copy of itself, it will continue to reproduce too with the virus. So that is how the lysogenic cycle takes place. So we are talking about this cycle under replication. That how do they replicate? So the major ways by which they can replicate is through lytic cycle and through lysogenic cycle. I hope it is clear now for the person that asked. Capsid is just the outer protective layer, nothing special. It is just the outermost layer that is protecting the virus. That is the capsid, the outermost protective layer that protects the virus. Please, is the person clear?